So since CAR T-cell therapies are mostly an autologous treatment, this treatment modality is based on or withdrawing T-cells from the patient's blood. Then the T-cells are further enriched, purified, and transduced to express the respective receptor, and in order to reach the respective therapeutic doses, the T cells are then subjected to an expansion phase, and once the therapeutic dose is reached, the T cells are then introduced into the patient's bloodstream, where they can exhibit their anti-tumor response. Now, in hematological malignancies, the application of CAR T-cell therapies has been a real success story ever since in 2011 the first clinical trials reported response rates of up to 93%, and ever since the FDA and EMA have approved 4 therapies in the treatment of different blood cancers. However, if we look at the solid tumor setting, the picture is somewhat different. Here, the clinical experience has been pretty disappointing, and although the very first clinical trials had been conducted in the solid tumor setting, there is not one approved therapy right now. So this clearly shows that our understanding of the working principles of CAR T cells in the solid tumors is still limited, and this is what we are trying to address at Miltenyi Biotec in the context of our research, that is to better understand the functional mechanisms of CAR T cells with solid tumors in order to improve this treatment modality further. And here we came to appreciate that we actually need to let go of the historic approaches, and that is to monitor a therapy's efficacy simply by looking whether a tumor signal goes up or down in the tumor, but rather gain a more holistic understanding of how the therapy behaves in vivo. What is its biodistribution? How does it interact with the tumor and also with the rest of the body? And for this, we acquired the MILab's optical imaging system in combination with the micro-CT modality. So the optical imaging allows for bioluminescent and fluorescent imaging in 2D, and when combined with the micro-CT modality, it is a full micro-CT, we can conduct different combination hybrid imaging such as BLT and FLT, in which the micro-CT data is used to optimize the reconstruction of the optical data. And what is of special interest for us is the combination of BLT together with FLT. So from these tomographic applications, what we can do is we can do in vivo longitudinal cell tracking of therapeutic cells and also their organ biodistribution, which is very important from a safety point of view. As for therapies against solids, we can do tumor and therapy cell dual imaging and quantification in the organs. And also we can analyze the cell status of the therapeutic cells. That is, where are the cells, where are they activated, and when do they start to enter into the exhaustion phase. And of course, we can quantify the injected dose and the development of cell proliferation over time. As regards for the exogenous agents, we can do PK/PD not only for oncology but also for other settings. We can do contrast agent quantification, nanoparticle drainage, and this all on a quantitative manner. So for our BLT-based cell tracking approaches, we used the following transgene design. We used a standard CAR design based on CD8 spacer and a 41BB and CD3 zeta signaling domains. The therapeutic CAR contained an scFv derived from a clinical antibody cetuximab, which is directed against EGFR, an antigen that is overexpressed by several tumors. And the negative control CAR contained an scFv directed against BDCA2. This is an antigen that is neither expressed by tumors nor in mouse tissue. And downstream of the CAR, we engineered click beetle red luciferase separated by a P2A element. So this P2A element induces autocleavage. One translation comes to this point, ensuring that both gene products are separately expressed but co-expressed in the cells. Our study outline was as follows. On day -11, we established tumors by a subcutaneous injection of the pancreatic tumor cell line ASPC1. Then the tumors were allowed to grow, and on day 0, therapy was started by an IV injection of the therapeutic cells or the respective mock T cells. And in a 2 to 3 days interval, we imaged the biodistribution of the cells. So here I have to say, in this study for the 2D imaging, we actually used the IVIS Lumina, and for the 3D imaging, we used the OI micro-CT modalities from MILabs. And what you can appreciate directly is that the tomographic images allow for better spatial resolution of our CAR T cells within the organs. Here for the 2D images, you see that with time at some point you get more scattering, the image becomes diffuse, and at some point there is also a certain degree of educated guess to narrow down where the T cells are located, while in 3D images you really, with an organ segmentation, can track this down. Now, as for the therapy, directly after injection, you see the immune cells going directly into the lung, which is pretty normal. And then after that, they start to distribute in the body. And for the control CARs, you see a extensive homing into the lymph, into the spleen. And after a while, this signal becomes more. And then later on, we see also passive accumulation at the tumor site. As for the therapeutic CAR, we actually see a different picture. So directly after injection, they too travel to the lung. After that, we actually see the very first signal popping up at the tumor region. And only after 13 days or so, a second signal comes up in the spleen, showing that you have also a circulation to the splenic side. And when we use the micro-CT to quantify the tumor growth, you see that in the control group, the tumor keeps growing, while in the therapy group, you have starting week 2, a tumor control. It drops in the beginning, but then it plateaus very quickly. And when we look at the BLT data within the tumor, it becomes also pretty apparent why there is tumor control in contrast to tumor eradication. So in the beginning, when the T cells recognize their antigen, you have a strong anti-tumor response. The T cells start to proliferate. In week 2, there is a certain plateau, and after that, the signal decreases. This is due to the fact that these T cells are heavily active against the tumor, but at some point, they reach a state where they go into an activation-induced cell death. That is, they're so overactivated that they cannot combat it anymore, and this clearly hints at there is room for improvement for this therapeutic modality, that we actually fine-tune the signaling capabilities. So I know this slide beforehand was pretty compact, so I selected 2 specific videos to show you in detail how the data looks like. So here we see the therapy CAR T cells. On the right side, you see a mouse that did not receive any treatment, while the others 2 did. And here, directly where the tumor was injected, is a signal, and also a splenic signal pops up. While when you look into control CAR T cells, here again, this mouse did not receive any treatment. This was actually a failed injection. And here you see a very nice tumor signal, and when the mouse then moves, you also see slightly the passive accumulation of the control CARs in the tumor. Now, such BLT-based tracking approaches are actually very nice in the research setting where you want to study new mechanisms. However, if you want to go one step further and use the data for clinical trial submission, you will most likely not be successful. And this is because the regulatory agencies usually require you to analyze the product as it is. However, if you introduce a luciferase into the cells, you by definition change your pharmaceutical product, which can have an impact on the biodistribution, the efficacy, etc. So as a result, we use fluorescent molecular imaging to image the unchanged pharmaceutical product over an extended period of time. And the contrast agents we are developing for this are antibody-based, and we couple them to hydrophilic dyes. This is a proof of concept study where we used mice injected with CAR T cells co-expressing luciferases that would also give us a BLT signal, and then we counterstain them with our contrast agents at specific time points. So as you can appreciate here, the therapeutic CAR comes up with a signal at the area where the tumor was injected, while the control CAR shows a splenic signal. When we compare it to the FLT data, you also have a signal in the liver, which is due to the degradation of the contrast agent, but also at the area to where the tumor is located. And when you do an overlap, you see that we have a nice co-localization of both signals, indicating that with these both modalities we can detect our CAR T cells. Now, as regards for the control CAR, we also have a splenic signal, as you can see here, by BLT. The reason why it is more pronounced by BLT is simply due to the biology. So we inject the contrast agent 24 hours prior to imaging. However, the T cells usually circulate between the spleen and the blood, so you usually have an attenuated signal in the spleen when using FLT. Still, it is present, and we can detect it via FLT. So what is also possible, and this was already mentioned, is by using the Imalytics preclinical software from Grimza IT, we can do full organ segmentation, and this way narrow down to where the cells are exactly located, in which organs, and at what quantities. So here we conducted the segmentation for all the mice and then looked at the signals. Naturally, for the BLT, you will see the biggest signal in the tumor and in the spleen, because this is where the cells are trafficking primarily. For the FLT data, you have also some degradational product going on. This is when you look at the untreated control. But when you compare to the therapy and the control-treated CAR T cells, you see additional signals popping up. That is in the tumor for the therapy CAR-treated group, and for the in the spleen for the control-treated CAR group, which is due to the localization and infiltration of the T cells in these organs. So this brings me directly to my conclusion slide. So longitudinal in vivo 3D imaging using the micro-CT BLT data has proven advantageous for us for the analysis of CAR T cell biodistribution, trafficking, and also persistence over so the presence of the CAR T cells from an extended period of time. Then when you want to move away from the research perspective to the non-clinical development, you actually see that the FLT-based applications are pretty advantageous, because you can directly use it for your trial submissions. And next steps are to further develop the BLT, FLT hybrid imaging for us. I have shown you an application that is the in vivo validation of newly developed reagents, but you can also go further and do tumor therapy, dual imaging, and other applications where you, so there are no limits to the imagination. And with this, I would like to take the opportunity to to thank many people involved in this project. So from the Miltenyi part, there were many colleagues involved with their input. One special thank you goes to Vael Aravashte, who actually set the stage for these projects, and Janina, Katrin, and Katarina, who conducted the experiments. A huge thank you goes to the MILabs team, who supported us with their excellent help and support and knowledge. And also a thank you to Felix Gremse regarding organ segmentation and the analysis of the data. Thank you very much for your great help, and thank you for your attention.